Hello students, this is Professor Chalai, and in this video I'm going to go over some of the things that you would need to know for the Bleach Kinetics lab. Now bleach, as we all know, discolors our clothing, right? So if we have some colored clothing or some colored dye, and in our lab we're going to use red 40 dye, interacting with bleach, it'll produce some colorless product. Bleach, in case you're wondering, is sodium hypochlorite. Okay, so this is our reaction. If we were to write a rate law for this reaction, it would look something like this. The rate of the reaction is equal to a rate constant K times the concentration of our dye, in this case it's red 40, to some power, let's call it A, times the concentration of our bleach to some power, let's call it B. Now, when we have something like this, we can actually simplify this rate law if we if we make one of these concentrations so large that it doesn't actually change during the course of reaction. So in lab this week, we're going to make the bleach concentration much higher than red 40 so that it doesn't really change, and so we can think of the bleach concentration as constant throughout the reaction. So if the bleach concentration is constant and K is constant, we can group these two things together into a single constant we're going to call kappa prime. So if we're going to group K and bleach to some power, we're going to call it a new constant, kappa prime. This will be a pseudo rate constant. It's not really a rate constant, but to help us analyze this rate law further, we're going to group those things together. So then we can rewrite our rate law in terms of this pseudo rate constant kappa prime like this. And that way we can only focus on red 40, at least for the beginning. Okay, so we have a new rate law here in terms of this new rate constant kappa prime. It turns out red 40 is a dye, has a color, it's red, so we can track its concentration over time by using absorbance. So we know if we plot or we track the absorbance of red 40 over time, we know that absorbance will be related to the concentration of red 40. Right, so we can track its concentration over time. As it gets destroyed, its absorbance will drop. Right, now we know using integrated rate laws that we can figure out the rate order and the rate constant from various time plots. For example, for a zeroth order reaction, we can plot concentration of, I'm just going to use R for red 40, concentration of our dye versus time. For a first order reaction, we can plot natural log of concentration versus time. And for a second order reaction, we can plot 1 over concentration versus time. And whichever one gives us a straight line, that will be the order of the reaction. Now, since we don't really have the concentration versus time, we have absorbance versus time, we're instead going to plot absorbance, natural log of absorbance, and 1 over absorbance, but the information that it gives us will be exactly the same. So if this plot is a straight line, then just to remind you of the full equation, right, plus the starting concentration, it means that negative the slope is K, or in this case actually kappa prime, and the order is zeroth order. If it's this equation, right, it means that negative the slope, I'll put kappa prime there, negative the slope is kappa prime, and it's a first order reaction. And if this plot is a straight line, then it means that, let me just finish this, it means that the slope is kappa prime and it's a second order reaction. So you will need to make all 3 of these plots to determine what the order is and what kappa prime is. So let's go back here. So we'll figure out what the order is and what kappa prime is. Once we do that, there are other parts of this equation we still need to figure out. For example, what is the rate, and what is K, and what is B? All right, so let's go through that. So we know that we can get an initial rate from negative the change in concentration of red 40 over the change in time. Negative because red 40 is a reactant, right? Red 40 is a reactant right here. So if we make a plot of concentration versus time, right? Negative the slope of that, so concentration versus time, right? Should be a negative slope, and the initial rate we can get from the slope. The initial rate will be positive, the slope is negative, so negative of that will give us the initial rate. However, we don't have a plot of concentration, right? Remember, we're plotting absorbance, but they're related to each other. So a plot of concentration versus time will give us that initial rate, which is a plot that we already made up here for the zeroth order. That's a concentration versus time plot. Except for this initial rate, we need to only plot the initial part of the concentration versus time plot. So if your concentration versus time plot looks something like this, or rather absorbance versus time plot looks something like this, right? We only need to plot the initial sort of the slope of the initial part of that curve, so you can delete the rest of the data and just plot the initial few points in this to get the initial rate. So that's how we will get the initial rate, which we can plug into this equation, right? So in that case, we will know the rate, we will know kappa prime, we'll know A, and we're ready to solve for K and B. So what we'll do is we'll repeat this experiment a few times. In each time, we'll change the concentration of red 40 and change the concentration of bleach, right? So we'll have a few trials, and for each we'll get the initial rate, and we'll pick two of the trials where the red 40 concentration stays the same, but the bleach concentration changes, right? And from that, we will use the initial rate and we'll solve for B, which is the rate order of B, which you've done before in our previous lab. So once we've figured out B, which is the rate order for bleach, the last thing that we need to figure out is what is the rate constant K. But to do this, we already know what the initial rate is. We already know what our initial concentrations of bleach and red 40 are. We know what our rate orders A and B are. So the only unknown here is to solve for K, and then we'll know each part of the rate law for this reaction. All right, some of the things to remember as we finish up this lab is bleach can stain your clothes, so wear clothes that you don't mind getting stained, or rather bleached, right? So wear appropriate clothing. And the second point is that the chemicals that we're using in this lab are red 40 and bleach. Red 40 is food dye. Bleach is used as a household cleaner, so technically we can wash these down the sink with running water. So for our waste, we can wash waste down the sink with running water. Another thing to think about is this is a kinetics reaction that we're measuring absorbance versus time. So being fast is critical, right? If you take too long to put your cuvette into your spectrophotometer, right? You're gonna miss this initial rate part, so you must be fast. So come prepared to, read your instructions before, because you'll need to be fast to run the reaction. All right, I'll see you all in labs. Bye.